Hi there. I'm Molly. Based in Oxford from the University of Oxford. So I'm in the UK at the moment. So my presentation is of a case that I saw as part of the medical team within the UK of a patient with polycystic kidney disease. It's also dominant polycystic kidney disease, but I'll just refer to it from now on as PKD. Unfortunately, this patient suffered several sort of hospital admissions and severe infections from this. And so it led me to kind of do a literature review and a discussion with the medical team looking after him about what sort of options he had for the management. So just a background, as I'm sure everyone's aware, so PKD is the most common inherited renal disease, but there still isn't a cure, and the surgical management options are quite flawed by their complexities and the complications from them. The evidence base for building management plans is quite sparse, and around 50% of PKD patients progress to end-stage kidney disease by age 60. The underlying cause of the disease isn't well understood at all. It's just known that renal function deteriorates as the cysts form and increase in number and size in early adulthood. So since there's no cure, the management's surrounded by thinking about limiting symptoms, complications, and delaying the treatment for delaying the requirement for dialysis. So my patient's of a 67-year-old man who I'll call Mr. X, and he presented with a fever, some cold sweats, and feeling quite unwell. So he had known polycystic kidney disease, so he monitored his inflammatory markers, so his CRP, on his phone app that he was able to access his blood test results, which is a system they use here. I'm not sure about in the US. So he'd had 2 similar hospital admissions within the same year with a similar presentation. So this just shows his first hospital admission was for an obstructive uropathy. So his ureters were obstructed due to his kidney disease, and this was 2 months before. And he presented with right-sided flank pain, and they fitted a stent to deal with this. 3 weeks later, he was then admitted again with the same presentation, and he had sepsis from the same problem. So they changed the stent again. So that's the yellow and then the green on this. The pink was this presentation that I saw him for. So again, as he presented, his kidney function declined again with his eGFR. So his kidney function, filtration reducing and his creatinine reducing. So upon this admission, the relevant findings for this case were he had severe abdominal distension. It was very firm. It was difficult for him to walk around because of the size of his stomach. He had an umbilical hernia, and he actually had separation of his stomach muscles from the severe force that was inside his stomach, the pressure. He had all the observations as well that fit in with him having a severe sort of sepsis infection. So just to review, to give a clear picture of the whole case, most of the medications he was on was actually related to the complications of his condition. So things like the increased clot risk. He had really bad acid reflux from a hiatus hernia that he had from the pressure inside his stomach, and he had reflux disease, and he was on tamsulosin for the symptoms he had. Since his diagnosis, 2 of his children have been diagnosed, and one of his children had been diagnosed and 2 of his sister's children had been diagnosed, as well as his sister. And prior to his diagnosis, he really enjoyed long distance cycling, but he was too fatigued and breathless now to be able to do this. So there was some imaging done in the hospital, and it was confirmed that there was no other source seen for the infection except obstruction of his ureters again, due to the size of the cysts that were on his kidneys and his liver. So for this admission, there was a significant increase with the management he had. So you can see the orange marker is the increase in renal function. So what the doctors actually did was they took out the stent and they put in a double stent. So they laid 2 stents next to each other within the ureters, which they admitted was done sort of out of desperation, because this was the 3rd time this had happened. And after the operation, he was comfortable, and yeah, his function increased. So there was a lot of discussion around what to do in terms of the management, and this sort of decision to lay the 2 stents came after discussion with the patient as well, and considering his options. So I just had a look into the literature for the different options available. So stenting is the main treatment for obstructive uropathy. It relieves the back pressure on the kidneys, and although it's minimally invasive, there is still a risk of bleeding and infection. And importantly, 80% of the patients that have a stent end up with permanent discomfort or pain when they're urinating, as well as increased frequency and urination for them. So it's not a pleasant thing often to have, and often the symptoms aren't relieved by medication. A study found that generally you get better improvement with stenting if you have a better baseline renal function and you have smaller kidneys. So this doesn't actually lend too well to PKD anyway. Also, stenting isn't curative, and as we've seen in this case, the cause of the obstruction remains and compression often rehappens. An alternate option is to remove the problematic kidney. Nephrectomy is considered in about 20% of patients with end-stage kidney disease, especially those who have infected cysts. This obviously decreases the abdominal mass, and for Mr. X, like, he had his symptoms of pain, infection, acid reflux. He was very short of breath on exertion, so this would relieve a lot of this for him. However, nephrectomy patients need long-term dialysis, and we can't forget to consider the sort of time commitment and reduced quality of life that involves. Also, considering the scale of a nephrectomy operation, you have to think about risk for general anaesthesia, and often patients with PKD have hypertension and barianeurysm risk potentially, which you have to think about. And in the case of Mr. X, he had quite a lot of cardiovascular risks because of his BMI, so because of his size related to his stomach. So then we discussed about possibly draining some of the cysts. So I reviewed a systematic review that compared like antimicrobials, so antibiotics, and also surgical treatments, that looked at whether cyst infections were better managed by either. And in general, it sort of showed that possibly long-term prophylactic antibiotics were advantageous, because often when you just treat with antibiotics afterwards, that's not dealing with any recurrences of the infection. But obviously this also isn't curative. Considering cyst drainage, sclerotherapy, which is a method that uses needles to aspirate the cysts, is an option. Catheter drainage, however, was found to be better for larger cysts, which is more relevant for PKD. However, this obviously introduces the risk of infection if you're puncturing cysts inside the stomach. So this just sort of highlights the fact that all of the management options do come with significant complications, and also there was a real lack of evidence when looking into this. A study that looked from 1948 to 2014 still only found 85 patients. So discussing kidney transplant, there's obviously a lot of psychological stress around waiting for a donor, but it removes the option of having to have long-term dialysis with just a nephrectomy alone. Some studies, interestingly, I found had shown that a native kidney size can actually reduce, and the kidney will regress if you put in a new kidney, which then removes some of the complications considering of having a nephrectomy. Alternatively, some studies show that people who can't have a nephrectomy could have an arterial embolization. So you remove the arteries that supply that kidney essentially, and that can create some abdominal space and relieve some of these pressure symptoms that Mr. X had. So I had quite an interesting discussion with the patient as well about how it was found he had polycystic kidney disease. How come he didn't know of his parents having it, and he was the one who raised the flag in his family that he had it, and they all got tested. So he actually explained that he had quite a psychological impact from this, and he'd seen his general practitioner for over 10 years, explaining that he was really concerned about his abdominal distension, but the GP just kept telling him to lose weight and said, that he should seek counselling for his health anxiety. And it wasn't actually found that he had this disease until he had this emergency presentation in the ED. So I've thought it would be interesting to research whether him being diagnosed earlier would make a difference, because I had this discussion with him and he asked me these questions. There are some trials that look at molecular therapies to target cyst growth, and they show that if you target well, some studies have shown that you have less mutations when you are a child compared to an adult, which then thinks you'd have less cysts and therefore it could be beneficial. However, most of these studies that look at the molecular therapies for cyst growth are sort of in their infancy, and it's only preliminary data. However, if there are larger sort of randomized controlled trials, then some of the ones which have been shown to reduce kidney volume could be brought into routine practice potentially, which would certainly deal with a lot of these issues we've discussed. I think finally, just we thought about the impact on family planning. Mr. X hadn't been aware at all, and he said, maybe people would like to be tested as like a pre-implantation, but this is quite contentious in the literature between doctors. But I thought it was something that was important to raise. So these are my conclusions. And if anyone has any questions, please let me know.
Fantastic. Thank you, Dr. Abbott. I'd like to know, for cases like Mr. X, what can we do to collect more data or do more research on this kind of topic?
So I think that was something that I found when looking into the literature. It's really difficult because a lot of studies actually exclude PKD patients when they're investigating some of these managements for kidney disease or for kidney problems, because they'll see it as a confounder. But obviously that means then that a lot of these studies that even I looked at had to say, well, you're extrapolating the data to even assume that this would work for PKD patients. So I think reducing this as an exclusion criteria and just having them as a subgroup within studies would be one option. And also I think just encouraging PKD patients, when they feel comfortable to join on to sort of data collection. I noticed at the beginning that there was the Pkd Registry, and things like this are obviously really, really beneficial because it is a rare disease. It would be very useful to be able to collect any data on the patients that are willing to give it.
Thank you. We had one other question. If you could just comment a little more on kidney reduction versus a nephrectomy, kind of what you saw in the literature as far as when to recommend either.
So I think it seemed to come across that nephrectomy, if possible, relieves a lot more symptoms for people and has better understanding in the literature, but also surgeons are more comfortable with these kind of procedures because they're more common than doing the things like the arterial embolization to try and reduce the size of the kidney. These aren't nearly as common or sort of well practiced. In general, the reason I looked into the kidney reduction is that for a lot of patients who have PKD, the general anesthesia risk of being put to sleep for such a big operation means that they're not considered. And when I spoke to the doctors that I was working with for this patient, they said that they didn't think that it would be safe for him to have a nephrectomy, which is why I just thought, well, I'll have a look at what the other options were. So I think in general, nephrectomy, if possible, is more preferred, was the general sort of impression I got. However, unfortunately for a lot of PKD patients, that's not considered an option.
Thank you, Dr. Abbott. And of course, we at the PKD Foundation want to encourage everyone to speak to your doctor about your own individual care. But it's really interesting to see a case study like that. So, thank you. All right, let's move on to Dr. Bland.
All right, I'm going to share my screen here. Oh, it's actually not giving me the option to.
Try now.
Okay. Oh, let's see here. Still not allowing me to. Here we go. All right. Okay. Can you guys see my screen?
Yes, we can.
Okay. Oh. Sorry, my PowerPoint just froze. Okay, can you see my mouse on here? Or did it move for you guys to the next slide?
Yes.
Okay.
Just move to the next thing.
All
We're
right.
back on the title page though.
Think we got it. Okay, well, thank you everyone for being here today. My name is Sarah Bland, and I'm a postdoctoral researcher at the University of Oklahoma Health Sciences Center. I have no disclosures to make today. So today I'll be talking to you about my project that's looking at a population of TREM2 positive kidney resident macrophages that are associated with slowly progressing cystic kidney disease. So cystic kidney diseases are multisystemic disorders and can have several different etiologies. There's many types of cystic kidney disease. These include AD-PKD, AR-PKD, Meckel-Gruber syndrome, and many others. They affect approximately 1 in 500 people and lead to cysts not only just in the kidney, but also in the liver, and eventually lead to end-stage kidney disease and liver fibrosis in patients. Currently, there's limited treatment options available, including tolvaptan and renal replacement therapy. Now, while these diseases have several different etiologies, one thing they all share is that all of these diseases are what's called ciliopathies. So this means that they're caused by mutations in genes that somehow affect the cellular cilia or ciliary function. So primary cilia in the kidney are these tiny little sensory organelles that are found on the apical surface of epithelial cells lining the nephron and the collecting duct. We still don't fully understand how mutations in these genes lead to cystic kidney disease. But one likely contributing factor in the progression of disease is the immune cells that are present in the kidney. Now, in the cystic kidney, there are a number of different immune cells present, including T cells, neutrophils, monocytes, and then the cells that our lab is most interested in, the kidney resident macrophages. So kidney resident macrophages have long been thought to be involved in cystic kidney disease. They're believed to promote cystic epithelial cell proliferation, thereby driving cyst expansion. They're also believed to play a role in extracellular matrix deposition, thereby promoting fibrosis in the kidney. And then lastly is tissue resident cells. They're kind of in a great position to affect both the infiltration and activation of other types of immune cells coming in. So kidney resident macrophages are therefore thought primarily to drive progression of disease and thus be detrimental in the cystic kidney microenvironment. Now, we know in humans with PKD, during the first 2 decades of life, kidney function is on average normal, and cyst progression begins sometime in the 2nd decade. Patients will typically not reach end-stage kidney disease until somewhere between the 5th to 9th decade of life. So in humans, this is a really slowly progressing gradual disease. Now, many of the studies implicating macrophages in disease progression were done using animal models with rapidly progressing cystic disease. So if we look at the table I have shown here, I've listed a couple of the different rapid disease models that we use in animals. Now at the top here, you see the Cpk Animal Model. This model would be equivalent to looking at the entire course of disease in a human between the ages of 0 to 5 months. So these rapid models are not necessarily homologous to how the disease presents and progresses in humans. There are slower progressing disease models, however, such as the CRE-IFT88 aged model shown down here at the bottom, and then the PKD1-RC model that progress much more slowly over the course of the animal model's life and are a bit more representative of what the disease typically looks like in human patients. So we wondered whether the macrophages that are present in these slower progressing disease models would have the same function as those that are present in the more rapidly progressing disease models. So today I'm going to be going over data from 2 different models, a rapid model, the CRE-IFT88 IR model that we use in our lab, and then the CRE-IFT88 aged model, which is the slower progressing disease model. Now, for these experiments, we began by inducing cystic disease in our mice by tamoxifen administration. So in one group of mice on the bottom half of this graph, 3 weeks after tamoxifen injection, they're given an ischemia reperfusion injury to the kidney, or IR injury. 2 months later, we collect the kidney tissue from these mice, and we end up with 3 different groups. So we have our rapidly progressing cystic group, and then we have 2 rapid controls. On the other group of mice, after tamoxifen injection, we let these mice just age and let the disease progress for approximately 7 months before our tissues are collected. And we end up with 2 groups of mice from this group of animals. We have our slowly progressing cystic group, and then we have our slow aged control. Now, after tissues are collected, we sort out the cell groups that we're interested in, and then we submit them for single-cell RNA sequencing. Now, after sequencing and processing the data, we use a program called Surrogate to generate what are called UMAP projections of our immune cell data. Now, what a UMAP does is it visually shows you the different groups that we call clusters that are present in your data, and each cluster is shown in a different color. Now, the clusters that I'm showing here are separated based on their expression of certain genes. So in the bottom panels, the UMAP is overlaid with a heat map to show expression level of the common markers we use to kind of delineate or define our clusters. So in the heat maps, the more pink color there is means the higher expression level of the gene that's referenced, whereas the blue means less expression of the gene referenced. So in our first panel here, where it says C1QA, this is a very common macrophage, resident macrophage marker. So we know that the clusters that have this hot pink expression are going to be our kidney resident macrophage clusters. So after generating our UMAPs for our 5 experimental groups, we noticed that there is one cluster that's much more abundant in our slowly progressing disease model. And this is cluster 4 that I've circled here in red. And this is a kidney resident macrophage cluster that we found was defined by TREM2 expression. Now, TREM2 is a gene that encodes for the protein triggering receptor expressed on myeloid cells. Now, while it looks more abundant in these UMAPs, we wanted to go ahead and quantify the number of cells as a percentage of all of the immune cells that we sequenced. And as you can see, we do see a significant enrichment of this TREM2 cluster in our slow disease model when compared with the rapid disease model. So this data was from our RNA sequencing, as I mentioned. Our next step was to verify these findings by a process called flow cytometry. Now, when we do flow cytometry, we use fluorescently labeled antibodies to mark our cells and determine what cell types are present. Unfortunately, TREM2 antibodies have historically not worked very well at all, and surface TREM2 is also very commonly cleaved by proteases, meaning that it can be difficult to identify these TREM2 positive cells by flow cytometry. So we instead use what's called a surrogate marker, which is simply another gene that's highly expressed in our cluster, but for which we do have a reliable antibody to define it. So in this case, we're using CD206 as our surrogate marker. And you can see by the heat maps that TREM2 and the CD206 encoded by the MRC1 gene have very similar expression patterns in our data set. So using our surrogate marker, we analyzed kidney immune cells from each of our 5 groups by flow cytometry. So shown here are some representative flow plots from all 5 groups with the macrophage marker F480 shown on the x-axis, and then our surrogate marker shown on the y-axis. And so the top box in these plots is going to be representing our TREM2 positive macrophages that are present. And as you can see in our slow model, we have a much higher amount of these cells in that top box in the graph as compared to both a rapid model and then all of our control groups. So we quantified this flow data as well, shown here in this graph, and we saw that there was a significant increase in the amount of these TREM2 positive cells as a percentage of our live single cells. And we also have some preliminary data comparing this slow disease model to other rapid disease models. And while we have low N numbers currently, we are still seeing on average more of these macrophages in our slow progressing model compared with all of our rapid progressing disease models. So what I've showed you today is that while the 2 types of disease models we use in animals, these rapidly progressing and slowly progressing, they share many similar characteristics, but despite this, the macrophages that are present in these 2 disease models appear to be comprised of different populations. So for our future directions with this project, we first want to see whether this trend that we're seeing of more TREM2 macrophages in our slow models holds true for other types of disease models. So we have a number of other rapid cystic models we'll be comparing, and then other slow cystic disease models we'll be comparing. And then we want to see whether TREM2, the gene, is required for this population of macrophages, and what happens to the disease progression if we delete this gene or if we get rid of it. So with that, I will take any questions, and I'd like to acknowledge and thank my lab. They're always great on our long experiment days, the research animals and our funding that makes this possible, and then the PKD Foundation and the PKD Connect organizers for inviting me to talk and putting this whole thing together.
Fantastic. Thank you, Dr. Bland. If I could start with a question. So you mentioned that when you're making these mouse models, you intentionally induced injury on the kidney in some of them. Could you talk about why you do that? So when you delete the cilia gene, so when we give the tamoxifen to our mouse model, it causes a deletion of a cilia-related gene. And when you induce injury in those animals following deletion of the cilia-related gene, it leads to a very rapidly progressing cystic disease. If you delete the cilia and just let them go, like we do with our aged, they still will get cystic disease. It just takes a lot longer time. So makes sense. And then in cystic disease, these TREM2 macrophages, could you just clarify, are these beneficial or detrimental to disease progression?
It's a really good question, and we're not sure yet. So in another slowly progressing disease, Alzheimer's disease, these TREM2 macrophages are beneficial. So they help restrict the expansion of the plaques, and they improve cognitive function when they're present. However, in obesity and cancer, TREM2 macrophages are detrimental, so they're bad. So we're really not quite sure how it's going to go in our future directions. We think that because PKD is slowly progressing, similar to Alzheimer's, they might be beneficial, but we're not sure. We have some animal models currently in the works, so we're really excited to see what it does.
And, once you get through these experiments in mice, what does it look like for human patients? When can they expect to see maybe something translated to clinical trials?
So the exciting thing is, if these macrophages are beneficial in disease, there is already an antibody in clinical trials for humans for Alzheimer's. So that stimulates TREM2. So it activates these TREM2 macrophages. So if these macrophages are beneficial, there is a potential treatment kind of already in the works that could potentially be used in patients. That's again, assuming these macrophages are beneficial. Now, if they're not, if they're detrimental, we could still kind of use that same plan of attack. So another type of antibody to get rid of these or target these macrophages specifically. And that could also be used later on clinically for patients.
Great. Well, we look forward to seeing the results of your work. Thank you so much.
Yes,
if there are any other questions for Dr. Bland, feel free to type them in and we'll put them in the end if we can. All right, so now our last presenter, Dr. Rossi.
Hi, everyone. Let's see. Okay, so can you see my screen?
Yes.
Okay. Hi, everyone. So I have nothing to disclose. So let me introduce myself first. My name is Charles Rossi. I'm an assistant professor in the Department of Pediatrics at the Icahn School of Medicine at Mount Sinai in New York. I did my PhD in Germany at the University of Heidelberg, and then I came to Mount Sinai and did my postdoctoral training. I subsequently stayed on as an instructor and then promoted to assistant professor last year. So my background is not in PKD, but it's more in glycosylation and studying metabolism and how they contribute to development and disease. But just this year, I was awarded a PKD Foundation research grant, and a Young Investigator Award. So now I'm testing what I've learned using Mannose, which is a simple sugar, as an antifibrotic therapy, and seeing if it could be used also to treat the liver issues that develop in patients with autosomal recessive polycystic kidney disease. So first, let me give you a background on what fibrosis is. So fibrosis is just it's excessive deposition of extracellular matrix into the interstitial space in the liver. So this is usually in response to injury. Whenever you have an injury, a liver injury, such as with a hepatitis infection, an autoimmune disorder, or metabolic disorder, what happens is a specialized cell in the liver, a hepatic stellate cell, becomes activated and initiates a program of proliferation, contractility, and deposition of collagen to try to combat whatever the injury is happening. The problem is when you have chronic liver injury, it's excessively doing this, and the liver will become stiffer as if it's progressive. Eventually, you'll develop cirrhosis and require liver transplantation. This is generally what's known for liver fibrosis. Currently, the only remedy to address this is to treat the primary disease, but there's an active effort to develop antifibrotic therapies, usually targeting the activated hepatic stellate cell. And while none are approved yet, there are some that are in clinical trials. So in our hands, we are using a simple sugar Mannose to try to develop as an antifibrotic therapy. And I hope as the talk goes on, you will be convinced of this. So how does this connect to autosomal recessive polycystic kidney disease? So I'm not going to give such a huge background on ARPKD because I'm not going to really talk about it today. The only thing I wanted to liken it to is how it may fit together with glycosylation. So glycosylation is the addition of sugars to proteins. And so PKHD1 is the gene that is involved for the majority of cases of ARPKD. And this codes for a protein called fibrocystin, which is a highly glycosylated protein. So this already connects ARPKD, at least in the liver, to, well, in general, to glycosylation. So really I want to focus on the liver disease that develops in ARPKD. And what you have here is you develop a type of fibrosis called a congenital hepatic fibrosis and bile duct ligation due to ductal plate malformations. And you can see that here in this ARPKD liver. And this arises because of an abnormal development of the ductal plate in the developing liver that normally would produce a biliary system within the liver, but instead they produce dilated bile ducts. And there could be downstream problems with this. So how does this, again, connect to glycosylation in general? So we are using, what I do work on in recent years is focusing on an enzyme called mannose phosphate isomerase. So this is a central carbon metabolic enzyme that catalyzes the interconversion of mannose 6-phosphate and fructose 6-phosphate. This is an intermediate of glycolysis, and mannose 6-phosphate is typically used for glycosylation. Because there's so much more fructose 6-phosphate in the cell than mannose 6-phosphate, generally the enzymatic reaction goes in this direction and goes towards producing glycosylated proteins. However, when you have a mutation in MPI, it could restrict the flow in this direction, and so you have a restricted glycosylation on downstream proteins. The remarkable thing related to ARPKD is that these patients also develop congenital hepatic fibrosis with ductal plate malformation. So, so this suggests that there is a glycosylation component that could be contributing to developing this disorder. The remarkable thing about MPI CBG is that it's actually treatable with Mannose. So if you give oral supplements Mannose, you can just bypass the biochemical defect and correct the hypoglycosylation. Here you're seeing serum transferrin, which is used as a marker for correcting glycosylation You can see in MPI-CDG patients that are untreated, they have a reduced level of this, but it's corrected when you give Mannose. Also, serum liver enzymes are corrected here, showing antithrombin 3 and albumin. But most remarkable is the fibrosis in the liver, here measured using a technique called FibroSpan, which measures liver stiffness. You can see that when the patients are put on Mannose, it reduces the liver stiffness. So this supports that Mannose can be used in the context of MPI-CDG as antifibrotic. So we wanted to model this in the lab. And so what we did was we used a zebrafish model for MPI-CDG. And you might be thinking, wow, zebrafish, why would you use that? Remarkably, zebrafish liver is strikingly similar to human livers, and it actually acts as a great model to study human liver disorders. So what we do is using what's called a morpholino oligonucleotide, we deplete the RNA coding for MPI, and we could see that in these animals with the MPI morpholino, they have a problem with their liver. Here you can see, because it's lit up in white, they have reduced glycosylation in these livers, and most notably is that they have 5 days post fertilization. So this is at the larval stage. They have increased thickness of their biliary structures. Because the morpholinos don't stick around too long, we also developed a stable genetic mutant of MPI. And in the heterozygotes in the adult zebrafish, so this is at 18 months, you can see that the fibrosis that they develop is remarkably similar to what looks like a congenital hepatic fibrosis with ductal plate malformation. And finally, if we look at the expression of fibrotic markers within the livers, we see that in these morphant livers, we see an increase in these fibrotic markers. So the next question is, okay, well, if we give them Mannose, can we alleviate this fibrosis? And indeed, we see that when we feed the morphins Mannose, they do recover. And if you look down here, you also see a decrease in expression of the fibrotic markers. So the next question we wondered was, okay, is this something specific to when you deplete MPI, or can this be possibly used generally as an antifibrotic? So to look at this, we first stuck with zebrafish, and we induced fibrosis by giving them 2% ethanol in their water, and with or without Mannose. And indeed, we saw that in the ethanol plus Mannose group compared to the ethanol alone group, we saw a reduction in the expression of fibrotic markers. So this sort of suggested that, okay, this is more general than simply when you have an MPI deficiency. It may be more generally used as an antifibrotic. So we wanted to move away from fish then and see, would this work in a mammalian system? Because ultimately we would want to use it in human patients with fibrosis. And so we used a carbon tetrachloride liver toxin model, a CCl4 liver toxin. And after 4 weeks of injecting CCl4 in their peritoneum, they developed liver fibrosis. And we would put them either on water, on normal water, or with Mannose in their water. And indeed, you can see with the CCl4 alone, they would develop this bridging fibrosis. You can see here, but when we included 5% or 20% Mannose in their drinking water, the fibrosis was remarkably reduced. And we could stain for this using collagen. And again, you could see the red staining is increased with the CCl4, but then when you add 20% Mannose, it's substantially reduced, and that's quantified over here. So it looks like Mannose in general is suitable at acting as an antifibrotic. So recall in the beginning when I said that the hepatic stellate cell is the main driver of liver fibrosis. So we wanted to know, is Mannose acting at the level of the hepatic stellate cell? And indeed, what we found was that when we, these are two different human hepatic stellate cell lines, when we cultured them in culture, we could see that just simply culturing them, these types of cells become activated. And when we added Mannose, these activation markers decreased. And this happened in a dose-dependent manner. So it looks like the Mannose is acting, and this was all published in 2019. We did a much more comprehensive analysis of this. So you can refer to the paper if you want to look more at it. But finally, what we wanted to know is what's happening to the glycosylation. So we're currently looking at the livers in the CCL4 model and another animal model that we're using. But when we looked in the hepatic stellate cells, we did using liquid chromatography and mass spectrometry to look at glycosylation of global proteins. One protein that came up was epidermal growth factor receptor. So you can see here, this is the control LX2, and in the MPI mutant LX2, you can see a reduction at, for example, this glycosylation site here. But when we would give Mannose, it would restore the glycosylation abundance at that location. Interestingly, EGFR has been tied to liver fibrosis, being involved with progressive liver fibrosis. And so we were very happy to find this. And this is something that we're focusing on more in the future to see if this potentially could be an important player in how Mannose is mediating its antifibrotic effects. So just quickly in summary, so we developed a zebrafish model for Mpi deficiency that mimics MPI-CBG in humans, including reduced glycosylation in the hepatic fibrosis with ductal plate malformation. Mannose mitigates the severity of fibrosis in MPI deficient zebrafish. And Mannose can also mitigate fibrosis in zebrafish and mouse models independent of the MPI deficiency. And finally, Mannose can reduce the fibrotic response in human HSC and restore EGFR glycosylation in MPI deficient HSC. And just very quickly, this goes on to how it ties into the PKD award that I received, because now we're expanding this research into an animal model for ARPKD. So using a PKHD1 knockout mice, we are going to treat it with or without Mannose and also use an EGFR inhibitor. This actually has been published in PKHD1 deficient mice to reduce the fibrosis in these animals. And we will look downstream by glycoproteomic analysis and histology and molecular analysis. So with that, I just want to thank especially Jamie and the rest of the people in the lab. And this is my contact information if any of you would want to reach me, and I'll be glad to have any questions.
Thank you, Dr. Rossi. So we do have one question about types of liver nodules. Can you comment on Mannose in follicular nodular hyperplasia?
Follicular nodular hyper. So you mean, is it effective?
Do you have any specific research on types of liver nodules with Mannose, or is this more general?
So I'm not familiar with those nodules, to tell you the truth. Are these pre-neoplastic or something? Or is this just, is this cysts?
Unclear. I might connect you afterwards with Stacy who asked the question.
Okay, so I don't know offhand that answer. Mannose has been shown to be somewhat beneficial for people with autoimmune disorders, not people, in animal models of autoimmune disorders, such as inflammatory bowel disease, because it seems to induce the expansion of regulatory T cells, which modulates the immune response. And it does this through a transformative growth factor beta mechanism, although they're not exactly sure how that works. So there are other disorders that this may be useful for.
What about kidney fibrosis? Is there any evidence that Mannose might have an impact on that?
That's a great question. And actually that's something, since I'm expanding into PKD now, it's something I would want to look at, because I would think it would be effective. So this is something I'll, keep an eye on as I'm running these experiments in the animal models.
And then also, if we're thinking about using Mannose as a therapy, are there any negative side effects that that you're aware of?
So generally, Mannose is considered very safe. It's just a simple sugar. You can buy it over the counter. There are some instances where patients maybe have some gastrointestinal bloating, especially if they eat a lot of it, because it's not taken up very efficiently by the gut. And so you have this, if you eat a lot of Mannose, it'll be a bunch in your gut, and you'll have, because of osmotic considerations. You'll have fluid go into your intestine. The other thing that you have to be careful of is if you have an MPI deficiency. Because when you have an MPI deficiency, you can't, MPI is important in removing any excess mannose. And so if you don't have, or if you don't have MPI there, it's an embryonic lethal, it's not compatible with life. If you have a low activity of MPI, you may get a buildup of mannose 6-phosphate, and this could have some detrimental effects. But generally speaking, mannose is safe, and it's already used to treat MPI CDG, and it's used to treat urinary tract infections in the clinic.